The circulatory system is a sterile environment, and the presence of any bacteria is generally significant. There are multiple ways that bacteria can enter the blood. This can include direct entry through procedures such as toothbrushing or from pre-existing infections that can disseminate into the blood. Contamination of the blood may be categorized as bacteremia, which is the intermediate presence of bacteria. Septicemia is a blood infection where there is significant amounts of growing bacteria circulating the blood. Sepsis is a life-threatening complication with three stages: sepsis, severe sepsis, and septic shock. A unique disease associated with sepsis is endocarditis, which is the bacterial colonization of heart valve leaflets. There are two types of endocarditis, acute and subacute. Subacute is often caused by the HEC organisms outlined here. The specimen received from infected patients is a blood bottle, which is a mixture of blood and additives to quickly flag for contamination. A gram stain can be performed on the sample for quick differentiation between gram-positive and negative organisms, while the specimen is inoculated on HbA, McConkey, chocolate, and CNA. Once the organism is identified, sensitivity testing must be completed following the UCAS method for isolated organisms, with antibiotics selected specific for the type of organisms, except for any obligate anaerobes, which are immediately treated with metronidazole.
The gram-positive organisms identified were all cocci. A catalase test was performed to determine whether the organism was able to catabolize hydrogen peroxide into water and oxygen. A positive result suggests a Staphylococcus species, which leads to the use of a coagulase test. This tests the ability to convert soluble fibrinogen in the plasma to insoluble fibrin. A positive result is a presumptive identification for Staphylococcus aureus, which can be confirmed by a DNase test. A positive result of the DNase test implies the organism can digest DNA, and this test is also an indicator of Staphylococcus aureus, which is a common cause of acute endocarditis. Comparatively, a negative coagulase result represents a coagulase-negative staph. An overbiasing disc is subsequently used for identification purposes by differentiating organisms that are sensitive or resistant to the antibiotic. However, a sensitive result requires the use of an API staph, which uses a series of biochemical tests to positively identify a Staphylococcus organism. The API staph was used to identify Staphylococcus epidermidis, which is a common cause of sepsis. Conversely, if the gram-positive cocci was catalase negative, an analysis of forced blood agar enables the differentiation of potential organisms based on hemolysis, with Streptococcus being the presumed species. Alpha hemolysis is indicative of partially hemolyzed red blood cells, while beta hemolysis is complete hemolysis of red blood cells. Gamma hemolysis indicates an organism incapable of hemolyzing red blood cells. An optogen test is then used to differentiate between alpha hemolytic Streptococcus. A sensitive result indicates Streptococcus pneumonia. The positive identification of Streptococcus pneumonia is supported by morphology of the organism, which is diplococci appearing as cations.
A resistant optogen result suggests Streptococcus viridans. As the presence of Streptococcus viridans is significant in the blood, an API strep should be used to specify which organism in this group is present, as it is responsible for subacute endocarditis. For those organisms that are gamma hemolytic, an ampicillin disc was used to differentiate between Enterococcus species. If the Enterococcus is sensitive, the growth on the MAC plate is further observed to identify the species. In conjunction with the previous test conducted, growth on MAC is a positive indication of Enterococcus facialis. This species is a cause of subacute endocarditis. A gram stain may also reveal a gram-negative rod, which percepts the use of a catalase test. If catalase positive, an oxidase test will be performed, testing for the presence of cytochrome oxidase in the presence of oxygen. If the gram-negative rod is catalase positive and oxidase negative, the plates are observed for anaerobic growth. A strict anaerobe is verified using a metronidazole disc, which is an antibiotic that is only sensitive towards strict anaerobes. Therefore, if sensitive, an API-20A is used to specify the genus and species of the anaerobe. Bacteroides fragilis is a strict anaerobe that was identified. All infections caused by strict anaerobes are recommended to be treated with metronidazole. Comparatively, a facultative anaerobe with growth on McConkey in conjunction with the previous results is indicative of an enterobacteriaceae and requires the use of an API-20E to identify the species. Note that Klebsiella bacteria with positive INZO biochemical tests can be identified as Klebsiella oxytoca, a common sepsis-causing organism.
A strict aerobe with growth on MacConkey has a number of further identification tests. If the morphology on horseblood agar appears as distinct green colonies, the C390 antibiotic disc can be used for identification. A resistant result confirms the presence of Pseudomonas aeruginosa, as this bacteria is the only organism resistant to this antibiotic test. An API-20NE was also set up to definitively identify and confirm this organism, as this API test is used to identify non-Enterobacteriaceae bacteria. For one strict aerobe with growth on MacConkey, a DNase test can also be used for its identification. A positive DNase result in this case indicates Stenotrophomonas species. Again, an API-20NE was performed to confirm the species, and this was identified as Stenotrophomonas multifilia. For other strict aerobes, an API test is needed to identify the organism. An API-20E was used, and this identified the bacteria Acinetobacter baumannii from a specimen sample. All of the strict aerobes that were identified today are common sepsis-causing bacteria. A gram-negative rod that is catalase negative and oxidase positive prompts the analysis of growth on the MacConkey plate. No growth on MacConkey in conjunction with growth on horseblood agar and chocolate agar in 5% carbon dioxide conditions provides a presumptive identification of Cardiobacterium species. The use of an API-NH was completed, which is used to identify Neisseria and Haemophilus species, however can also be used to identify other bacteria. Cardiobacterium hominis was identified, and this was supported by the strict plate growth conditions observed for this fastidious organism. This is a Hayseck organism and commonly causes subacute endocarditis. The following lab and doctor's reports are an example of one organism that may cause infection in the circulatory system. However, the blood is a sterile environment, and any organism present is considered significant and can cause disease.